Hey, welcome everyone to the week one session of Introduction to Cell Biology course. This is a PMRF TA course where we'll be discussing previous year's assignment questions and try to find answers to them and see if you guys have any other questions as well. My name is Ashwant and I'll be one of your PMRF TAs for this course. Okay, so we'll jump right into it. Last week, for the week one quiz, the first question that was asked was in the following figure, which pathway for the flow of information has not been discovered yet in eukaryotes? So the pathways are numbered numerically in red numericals here. So one is DNA to DNA, which would be replication. The other one is RNA to RNA, which would be RNA interference and RNA inhibition and so on and so forth. Uh, this is information flow, right? So it can be various regulatory mechanisms as well. The other one is protein to protein, which is obviously, as we might all know, is very possible in biology, where proteins regulate the function and activity of other proteins. So kinases, they can add phosphorus groups to proteins, either inactivate them or activate them, and so on and so forth. The other one is RNA to DNA information flow, or protein to RNA information flow, or DNA to protein information flow, or DNA to RNA information flow, protein to DNA information flow, or RNA to protein information flow. And the only pathway that has not been discovered yet in eukaryotic cells is DNA to protein information flow. So there is no direct effect of DNA on proteins, whether it is their function, their activity, so on and so forth. So we do not know if there is any direct information flow between DNA to protein in case of eukaryotes. Okay, okay. So the first accurate model of DNA was proposed by whom? Maurice Wilkins and Rosalind Franklin, James Watson and Francis Crick, Linus Pauling and Robert Corey, James Watson and Rosalind Franklin. So even though the first accurate model of DNA was proposed with the help of work from Rosalind Franklin and Maurice Wilkins, it was mostly proposed by James Watson and Francis Crick, right? And if you guys know enough about enough biochemistry, you would recognize Linus Pauling and Robert Corey in their significant work in understanding protein structure and folding. So James Watson and Francis Crick are the major players of producing first accurate model of DNA, which is this double helix structure that is supported by this sugar phosphate backbone, where the base pairs are interacting with each other with hydrogen bonds and so on and so forth. Okay. Does DNA dependent DNA polymerase make mistakes while replicating DNA? Yes, with an error rate of one mistake per thousand to 10,000 nucleotides, or no, the replication machinery is exquisitely refined by evolution not to make any mistakes. So it's a fairly straightforward question. So, if the DNA-dependent DNA polymerase is exquisitely refined by evolution not to make any mistakes, there would not be any evolution in the first place, right? So, if it is so refined that does not make any mistakes, there is no chance for mutations during replication. There's no chance for error during replication, and as such, mutations would not arise, and there is no evolution per se as we understand it. So, and in fact, we find that the DNA-dependent DNA polymerase makes mistakes, and the error rate of the mistakes is one mistake per thousand to 10,000 nucleotides, depending on the species, organism, individual, and so on. Okay. So the next question reads: What is Chargaff's rule for DNA? What is Chargaff's rule for DNA, and what does it state? So is it that there are twice the number of purines to pyrimidines in DNA, or is it adenine and thymine form double bonds and guanine and cytosine form triple bonds, or is it the fact that the ratio of purines to pyrimidines is always 1:1 in DNA, Or is it the fact that there is no relation between the number of purines and pyrimidines present in DNA? So this is one of the core principles of Chargaff's rule. So Chargaff's rule, in fact, states that the ratio of purines to pyrimidines is always 1:1 in DNA. And there are a couple of rules. So rule one states that the amount of adenine roughly equals the amount of thymine, and the amount of guanine roughly equals the amount of cytosine, which would imply that the amount of purine is roughly equal to the amount of pyrimidine. So that's why the ratio of purine to pyrimidine is 1:1. But there is also a second rule which states that the amount of adenine plus thymine is not always equal to the amount of guanine plus cytosine. Adenine plus guanine is equal to thymine plus cytosine, but adenine plus thymine is not equal to guanine plus cytosine. And this is very species specific, individual specific, right? So there could be more GCs than ATs in certain species or individuals. There could be more ATs than GCs in certain species and individuals. And as such, this ratio varies among different organisms, same in the but it's always same in the different tissues of the same organism. Okay. So what did X-ray diffraction pattern of DNA produced by Rosalind Franklin suggest to Watson and Crick? So as I was saying, Rosalind Franklin and Morris Wilkins helped in producing this first accurate model of DNA. But Rosalind Franklin was involved in producing the X-ray diffraction pattern of DNA, right? So what did this pattern suggest? Did it suggest that DNA is made up of purines and pyrimidines, or that it has a double helix structure, or that it has a phosphate backbone, or that it has a triple helix structure? Now, you cannot, X-ray diffraction is a physical diagnostic method. So you cannot really understand the nature of the chemical bonds that are present in through X-ray diffraction. I mean, you have techniques to do that. So like Raman spectroscopy and so on and so forth. But diffraction patterns generally do not do that. So the only thing they can one of the things that they can successfully tell is the structure of the molecule. So in this case, it was able to show that DNA is a double helix structure, but not only that, right? So we were also able to show that there are twists every 34 angstroms. There are 10 base pairs per twist. Uh, and we also know that DNA is a double stranded structure, and we know that the phosphates are on the outside, not the inside of the DNA. Okay. Which of the following statements about DNA replication is not true? Okazaki fragments are found on the lagging strand. DNA polymerase replicates DNA from 5' to 3' direction only. DNA helicase unwinds and separates the two strands of DNA. DNA replication is continuous on both leading and lagging strands. Okay, straight away, DNA replication is not continuous on leading and lagging strands, right? So that's why we have these Okazaki fragments. And what are Okazaki fragments? So if you look at DNA replication, the replication on the leading strand goes from 5' to 3' direction, which is going to go straight forward with the DNA polymerase. So it does not need to have to break. But when it comes to the lagging strand, which is going from the 3' to 5' direction, the DNA polymerase can only replicate in one direction, which is 5' to 3'. So it has to do it in parts. So it keeps doing it in 5' to 3' for some time, jumps back 5' to 3', jumps 5' to 3'. And because of this, there are these fragments of DNA that have replicated. These are called Okazaki fragments. And that's why it's discontinuous on the lagging strand. So that's why DNA replication is called semi-discontinuous mode of replication, because it's on the leading strand it's continuous, but on the lagging strand it's discontinuous. Okay, two strands of DNA are joined by what? Is it a phosphodiester bond, a covalent bond, a hydrogen bond, or none of the above? Uh, Fairly straightforward. So, hydrogen bonds are what help in joining two strands of DNA together. So, these are hydrogen bonds between the nucleotides that form the base of the DNA. The phosphodiester bonds are really present here on the phosphate backchain, and covalent bonds are basically there. And the only thing that binds the two DNA strands together are the hydrogen bonds. And adenine and thymine form two hydrogen bonds, whereas guanine and cytosine form three. Okay. Which enzyme joins the Okazaki fragment on the lagging strand? Is it DNA dependent DNA polymerase, helicase, primase, or DNA ligase? It is not DNA dependent DNA polymerase because that's what is doing this Okazaki fragment production and replication. Helicase unbinds the DNA, primase initiates DNA replication, and the only option that's left is DNA ligase, which joins the broken fragments in DNA, right? So, and you add the DNA ligase and PCR as well for this reason. So, yeah, DNA ligase is what joins Okazaki fragments on the lacking strand. Okay. So the next question reads, slightly numerical question. If a single molecule of DNA undergoes 5 rounds of DNA replication, how many molecules of DNA are created which do not have any original strand of DNA? So there are. So how do we know how many molecules of DNA are produced after n rounds of DNA replication? So that would be 2 to the power n. So the number of uh, strands of DNA that are produced after n rounds of DNA replication are 2 to the power n. So if it's 5 rounds, it would be 2 to the power 5, which is 2 times 2, 4, 2 times 2, 4, 4, 4. 4 fours are 16, 2s, 32, right? So if 32 strands of DNA are produced after 5 rounds of DNA replication, how many of these are newly created and do not have any original strand? So DNA follows semi-conservative replication. So the two original strands would remain two towards the end of the replication. So then the answer would be 32 minus 2, which is 30. Okay. So a nucleotide is composed of what? Phosphate plus sugar, phosphate plus base, phosphate plus base plus sugar, or none of the above. So if we go back to the structure of the DNA, the nucleotide is essentially composed of phosphate plus base plus sugar. So you have the phosphate group, which is the backbone. You have the pentose sugar, which links the nitrogenous base to the phosphate group. So together, these three are basically what a nucleotide is, right? Okay. So that's the end of week one questions. So